All right, so welcome back. And now we have the first talk by Achishman Raju, who's a faculty member of the Simon Center at the National Center for Biological Sciences in Bengaluru. So Achishman did his PhD from Cornell and followed it with a postdoc at Rockefeller, and he joined NCBS about 3 years back. So again, Achishman's visited us already before, and we had a very interesting set of discussions on the connection between statistical physics of emergent behavior and pattern formation in biological systems. So it's with great pleasure that I invite now Achishman to give his first talk, which is, I understand, will be a pure blackboard talk.
Thanks. Thanks, Sitabra. And so my first talk today is going to be a pedagogical talk. And, so I just want to get a sense a little bit of the background that all of you already have. So if you could just tell me by a show of hands, how many of you are undergraduates? Okay, so about half. And then how many of you have a biology background? Okay, so good proportion. How many of you have physics or a math background? Okay, so so my job is very difficult, so please sympathize with me. Half of you are physics, half of you are biology and math, and if there are other fields. But what I'll do is I want to sort of today set up some of the ideas that I will use tomorrow in my research talk, and I'll try and start with some very basic ideas, just so we are on the same page. And then towards the end, I want to talk a little bit more about, a more abstract viewpoint to think about the same ideas that I'll first discuss in an introductory way. So I have an hour, right, Sitabra? I have an hour, is that right? Okay. Okay. So, the first, so I'm going to be talking about, so you heard from Amitabha about morphogenesis. And typically in development, there are different things you care about. So morphogenesis itself derives from change in shape. But what I'm going to be talking about is not so much about morphogenesis as such, which is the change in shape, but I'm going to be talking about the fates of cells. And so I'm going to start by saying, what do I mean by cell fate? And essentially the viewpoint that I'm going to take is one that comes from a field of mathematics known as dynamical systems theory. And I want to show you how there is a connection between biological concepts that have come from the field of experimental embryology and developmental biology in general, and mathematical concepts that come from the field of dynamical systems theory. So I want to show you how there is how these two come together. Many of the same concepts have sort of different terms in these two languages. And the first thing, as I said, I want to talk about is the fates of cells, the fact that cells end up taking different fates in your body. So there are many types of cells. There are blood cells, skin cells, neurons. And what we are interested in one of the things we are interested in during development is why different cells end up becoming different types, and how we can understand this in what I'm going to be talking about is how we can understand this in a mathematical framework. So the first thing that I want to discuss is the concept of a cell state. And there are many views you can take on how to talk about the state of a cell. So for example, you could try and think about the state of a cell in a morphological way, right? Cells can be circular, they can be spindle-like. Cells come in all types of shapes. That's not something that I'm going to be discussing today, but in principle, that's an approach you could take. You could also take a sort of physiological approach where you really say that, I'm going to try and investigate the state of a cell by trying to see its response to perturbations. So in the case of microbes, I'm going to look at how they respond to drugs. In the case of say, or you could talk, how they respond to antibiotics, in the case of cancer cells, how they respond to drugs. And so, you could take a more physiological approach to cell state and just think, look at the response of cell to external perturbations. What I'm going to broadly take, though not quite, is that I'm going to accept the idea that there is a molecular basis to the state of a cell, right? So, I'm not going to be investigating all aspects of cell state, but I'm going to be talking about, what the, or at least I'm going to accept the idea that there's some molecular basis for cell state, which means essentially that we are interested in gene expression. And that has two parts. Right, so essentially in development actually, so you have, of course, you have DNA in your body, but DNA itself is actually quite boring when we are, when we think about development. Really, where all the interesting stuff happens, as Amitabh also showed you, is at the level of proteins. And so we are interested in the expression of genes. We are interested in what the proteins are doing as you go through embryonic development. What you're interested in is to figure out how do you now write a mathematical theory. If suppose in principle you have many proteins in your body, if you could write down, how do you write down a mathematical theory for how the concentration of proteins, or if you're interested in studying at the RNA level, how the concentration of RNA is changing as a function of time. So I'm going to be discussing how cell state changes, and essentially I'm going to be talking at the level of gene expression. So I'm going to, for this purposes of this talk, just say that all the changes in cell state that I'm interested in are given if I can track the changes in the state of the protein concentrations in the cell or the RNA concentrations, if I can track the change in gene expression. Okay, so a related idea to cell state, which I've already mentioned, is the idea of cell types. Okay, and as I said, you can have different types of cells, right? Skin cells, blood cells, fibroblasts. I mean, you could list out, there are about order of 100 types of cells in the human body. We're interested in the fact that at the molecular level, you have many proteins. Those somehow end up giving you distinct types of cells, right? And you really have, it seems like the types of cells in your body are in discrete, you only have certain types of cells in your body. If you, for example, make perturbations by knocking out certain genes, blocking their expression, you don't get new types of cells. What you do is you permute between known cell types. So this is sort of a very interesting fact about development, that even though cell state is determined through some very complex molecular basis, at the level of the phenotype, at the level of types of cells, there seem to be a few distinct types of cells that you get. And we are interested ultimately in understanding how that emerges. Okay, so as I said, we are interested in the change of a state of cell, and typically we are going to think of processes like this. Okay, I mean, so I'm just making up stuff here, but essentially you have different pieces of DNA, right, which I'm calling G1, G2, G3. These transcribe into RNA, which translates into protein. These proteins, if in particular they're transcription factors, so we're going to be in, that's one way of how you can regulate gene expression, can then activate, in this case activate, they could also repress the transcription of another piece of DNA, another gene, and which leads to a different protein, right? There could also be other things going on. For example, proteins could get phosphorylated, and for example, this protein maybe controls the rate of phosphorylation of this protein. So there are all interactions of this form possible. First, at the level of the transcripts, the fact that proteins can regulate the transcription of genes, and second, interactions of proteins themselves, which can also be regulated by other proteins or processes like phosphorylation and so on. And so typically you will see these kinds of diagrams whenever we discuss the change in gene expression, and there are many names for these. They're sometimes called molecular circuits, and if for a particular decision, particular developmental decision, you can identify the molecular players that are involved, then the idea is that you should just write down equations for the state of P1, P2, P3, maybe you want another variable for this one. So this is your set of dynamical variables. These will change as a function of time. These are the concentrations of proteins, and you want to write down differential equations for the change in these concentrations as a function of time, right? And you already saw a little bit of this in Amitabh's talk. Okay, so that's the basic setup, and let me now just discuss a couple of examples for how you might do this by taking some very simple examples first, so we're on the same page, before I go into thinking about this more abstractly. Okay, so let me take my first example. Which is that you have a single gene that you're studying that gives rise to a protein which can degrade, right? And so the differential equation you would write down for this is something like this, right? You have the rate of change of P1 as a function of time that has a production term, and there's a degradation term of the protein. Proteins naturally degrade. In fact, it turns out that the way the proteins degrade, there's an effective degradation of proteins because cells are growing, and in fact that turns out to be the major contribution to this degradation term. But in general, you will have production terms and you'll have degradation terms. So here, for example, if I had more players, I would write down differential equations for them. They would essentially involve production terms and degradation terms. Okay. What we are going to be most interested in our discussion is the steady states, right? When do these equations reach a steady state, right? So in particular, you have a steady state here when dp1 by dt is equal to 0, which means p1 of t is now just a fixed quantity which does not change with time, which I'm going to call P1 star, right? And this P1 star here you can easily calculate. P1 star is just equal to alpha by gamma. Okay, so these steady states we're going to call they're called fixed points, and we're going to be very interested in these fixed points. If I have the dynamics, if I have changing dynamics of proteins, there are certain points of the dynamics where you no longer get change in the protein concentration. These are called fixed points, and we're going to be interested in looking at them as we go forward. Okay, so the way to think about these fixed points actually is, and what I'm going to do throughout this whole talk is to try and take you away from thinking about particular equations. So unlike in and as a sort of broader philosophical idea, in physics, what you're often interested in is solving known equations of motion, right? And so in physics, you're depending on whatever the particular system that you're studying, the equations of motion are seen as fundamental. they're seen as something that you know, and then you apply them to different situations and you want to solve particular equations of motion. Here really the philosophy that we want to take is that the equations of motion are not really known, right? You can make approximations to what you think the equations of motions are, like we made here, but you know, we don't, this is sort of really, and it'll always be a phenomenological model of what the equations are, right? We don't really know, and in very few cases actually has anybody tried to quantitatively test equations at the molecular level, but we have some ideas of what these equations might be. So the approach that I want to take is that to not focus on particular equations, but rather take what I'm going to call a geometric approach, which is to try and think about the qualitative dynamics without thinking about, you know, the precise quantitative dynamics. And I'll show you today and tomorrow also in part of my talk why this, you know, change in approach, where you're no longer thinking about quantitative equations of motion, but thinking more about the qualitative, you know, their qualitative nature, can be very, very powerful. So this is you'll see as we keep going forward how we have this sort of change in approach, so to say, where you're going away from particular equations to more qualitative natures of the behavior of these equations. Okay, in this case, right, one way to visualize sort of more visualize what these equations are doing rather than try and necessarily write down what the equations are is of course just to draw these two functions, right? So here I have P1, here I have alpha, and here I have gamma P1. So this function is gamma P1, this function is just the constant function alpha, this axis is P1, and essentially what this equation is telling me is that my production rate happens at a constant rate, my degradation rate actually changes with P1, it's linear in P1, and there is a certain point where my production matches the degradation, where I'm going to get a fixed point, and my protein concentration is going to be this value P1 star. Okay, so another way to think about this is to draw what's called a flow diagram. Okay, so my flow diagram, so what a flow diagram is essentially it tells you that if I start at any value of P1, what direction will I move in and what will be the magnitude of that movement. So if I start off at P1 equals 0 in these equations, right, initially my production is going to be much more than my degradation, so I'm going to move in the direction of larger P1. So I'm going to move in this direction. Maybe I'll make a longer arrow. Okay, now I'm somewhere here, again my production is larger than the degradation, so I keep moving, but the magnitude of how much my P1 increases is actually becomes smaller and smaller till I reach near the fixed point where it goes to 0. If I start from the other side of P1, if my P1 is larger than this fixed point, then my degradation, as you can see, is larger than my production, and so my P1 is going to decrease. And so if I start from the other side, I'm again going to flow towards this fixed point P1 star, which we already calculated. Okay, so in this very simple example, I've already introduced a few concepts. There's this idea of fixed points, this idea of a flow diagram. In the physics literature also sometimes, they're also called phase portraits. It's effectively the same thing. I'm not going to be super careful about making sure again about the magnitudes and so on. Really, as I said, we're going to be interested in these fixed points, and we're going to be interested in the direction of flow to these fixed points. Like in this case, if we start from low concentration of protein, what this equation is telling us is we'll flow towards higher concentrations till we reach a particular point P1 star, which is a fixed point. In this case, if I have a higher concentration of protein, I still return to it, so this is called a stable fixed point, right? Whether I'm lower than P1 star or greater than P1 star, I return to P1 star. So this is called a stable fixed point. Okay, so hopefully this sets up some of the concepts that we are going to be using. So the next concept that I want to introduce here is the idea of a qualitative change in the flow, right? That here in these equations, if I change the values of gamma and alpha a little bit, actually, my flow diagram and everything else is going to sort of look the same. The only thing that's going to shift is the value of where P1 star is. So we are interested in more in changes to the flow which are more interesting than that. Okay. So actually, let me also pause here a second and again relate this to our discussion of cell types and cell state. Right. So remember, cell state I said was that if I take a bunch of proteins and I specify their concentration at a certain point of time, that is going to give me the state of a cell. What I'm going to think of as we move forward is these fixed points, these stable fixed points, as types of cells. Right. Because if you're near the fixed point, you return to these stable fixed points. These stable fixed points are sort of like cell types. They are, even though cell states take continuous value, there's only a single value of P1 star, right? It's just one value, it's a discrete thing, which corresponds to the stable fixed point. And so you can see already the analogy between, ideas of cell state and cell types, and here ideas of, the state as specified by a certain concentration of proteins and a type as specified by a stable fixed point, where the concentration of the protein doesn't change. Yeah. 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 Yeah.
So there are multiple fixed points.
Yes. 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 Yeah. Yeah. So we're going to come to that. Yeah. So we'll come to that. Okay. So that's great. So that's transition for the next part, where I'm going to introduce an example slightly more complicated than that one. So again, I have my production. And I have my degradation. But now I'm going to say that the production, right, the transcription of this gene that gives rise to this protein is regulated. So it can be regulated by this protein itself, and it can be regulated by some external signal. Okay, so I'm thinking of equations like these. So here I have an external signal I'm calling alpha E, which controls the production. Then you have this f of P1 term. Depending on the concentration of the protein, the production term can change. Then finally you have your degradation term as before. Okay, so again, as I said, I don't want to talk about specific terms, how you would want to model, for example, f of P1, but let me mention it. Usually when people want to talk now about the regulation of transcription, right, they will use forms for these functions that essentially come from sort of early 20th century chemistry. And so, a popular choice is Hill's functions. But again, I want to emphasize that in very few cases, and the best known case is the lac repressor in E. coli, but in very few cases have people actually quantitatively shown that these expressions are, right, these changes are governed by particular functions. But people use these because people think that they qualitatively represent the change of dynamics. And so again, so we are going to keep our discussion qualitative. So instead of writing out the function, I'm just going to draw it out for you. So this f of p1, we're going to assume looks something like this.
Excuse me.
Yeah.
Here.
Yeah.
Can you again explain what f of p1 was doing?
Yeah, so f of p1 is essentially saying that my p1 protein could be, for example, a transcription factor. You know what transcription factors are?
Yes.
Okay, so it could be a transcription factor which could then regulate the transcription of this gene. So whether this gene is transcribed or not, whether it leads to mRNA and then protein or not, depends on the current concentration of P1 in my system. So that's why dP1 by dt, the production of P1 is controlled by the current concentration of P1. And how it is controlled is what f of P1 is telling us. How does the concentration of P1 control the transcription of G1? Yeah, auto induction, yeah, exactly, yeah, I mean what you would call auto induction and auto regulation, this is modeling that. Okay, so yeah, exactly, thanks. So you have a gene that regulates its own activity, it degrades it, but its activity can be controlled by an external signal in this case. And the external signal is going to be important, and it's again going to introduce something that we are going to talk about more. So the external signal is important, I guess I should mention now, is because external signal is something that you can control, right? The external signal could be a hormone, for example. So in fact, this model has been, it's not something I made up, it's something that was used for the maturation of oocytes in frogs, which can be controlled by an external hormone. So it could be a hormone, but in other contexts, you could imagine some protein that you're overexpressing and so on, signaling protein. So all of these could be examples of external signals, but the important thing is that you can control this external signal and you can change its value. Yeah. Time delay. Oh, here. Yeah. No, that's a very interesting point, in fact. And there have been cases where these time delays become very important. Here, I'm not assuming any time delay, but you're right. There may be cases in which actually there could be a delay in transcription, and those delays can become quite important.
Sure. I mean, because it will take time for the protein to transcribe and then, translate and then affect the.
Yes. So, yes, I mean, the idea, you're right. I mean, so in a certain sense, you should really say that this. So there are two parts of this, right? Two parts to your question. The first thing is you could ask me, why didn't I include the mRNA, right? And in principle, I should have looked at, the RNA. The RNA itself can also degrade, and then the RNA can be translated. So why not include an equation for that? I could have included an equation for that. Most of the times, people don't do it because the dynamics of RNA are typically faster than the dynamics of proteins. RNA degradation times are much faster, and so they try and subsume all of that dynamics into just the equations for the proteins. But you're totally right that if you really wanted, then you should have had a variable for the RNA, you should have had another equation for it. There are cases in which the time delays don't affect things qualitatively. There are cases in which they do affect things qualitatively. Here I'm not going to be talking about the time delays, but they're very interesting, and it turns out the delay differential equations, which is what you get if you explicitly include time delays, are far more complicated than ordinary differential equations. And doing mathematics there is much more complicated also. So here I'm just going to discuss ordinary differential equations, which means the change only depends at the current concentration time. Yeah.
Thank you.
Is it something like with the increase of time,
Yes.
the feedback is more rigorous?
So we're going to analyze this exactly, what is happening, what this feedback is doing, and with increase of time, what happens to P1. But say the last word again, with increase of time, feedback is more?
Rigorous, or like
What does that mean?
what should I say? Will be more effective.
Would be more effective. So you are right that as the P1 concentration increases, the effect it has on the change in P1 will also be more.
Yeah.
But this cannot indefinitely happen. And so for that, this is why we need to specify a function f of P1. And the f of P1 that I've drawn here essentially eventually saturates. And the reason it eventually saturates is that you cannot infinitely increase the speed of transcription. Eventually, it doesn't matter how much protein you have, you're going to reach physical limits on how quickly you can transcribe. So you can't just keep increasing things, and right. So what you were saying then eventually would just take things to infinity. But in this case, you encounter certain limits to just how fast you can transcribe, and so eventually f of p has to saturate. And so again, the qualitative form of the function, no matter what it is quantitatively, has to essentially have some saturation term and something that increases before it saturates. Yeah. Okay, so going back to the signal alpha E, the reason this alpha E is important is it's something that we can change, right? So in an experiment, the alpha E is something that you can imagine changing. Concentration of a signaling protein, it could be cytokines in another context, it could be hormones in another context. We're not going to be very specific, as I'm not being specific about which genes these are and so on, because we want to set up a more abstract framework that's applicable to different situations. Okay, so as I said to you before, what we're interested in is in the fixed points of this. When does the dynamic stops? And that happens in this case when, so for a fixed point, when f of P1 is equal to minus alpha E plus gamma P1. So at the point P1 star where this is equal to this term is when you're going to get a fixed point of the dynamics. Now really maybe I should have kept these two together. These are the production terms, this is the degradation term. I'm only doing this for mathematical convenience, right? So I've just moved alpha E to that side, but it's the same thing as saying alpha E plus f of P1 star should be equal to gamma P1 star. The reason I moved it to this side is that you can recognize the right hand side is an equation of a straight line, right? It's just gamma P1 star minus alpha E, a constant, right? So it's easy to draw. So f of P1 star I've already drawn here. So this is f of, sorry, not P1 star, f of P1 I've already drawn here. This is f of P1. At alpha equals 0, I can draw a straight line, and I'm going to choose a particular slope. Right? Since alpha E is 0, at p equals 0, this right hand side has to be 0, so it has to pass through the origin. So this is just the line, let me call it 1, where 1 is just gamma P1. Okay, so again, for the fixed point, we need to solve this equation. I'm just solving this equation graphically by plotting the function on the right hand side and plotting the function on the left hand side. Right, what you can see already is that this example is more non-trivial than our first example, right? In the first example, we just had a constant production and degradation, right? The degradation, when it matched the production, you got a single fixed point. In this case, because of the way I've chosen f of P1, there are actually 3 fixed points, right? So I have a fixed point here. When things match, these two rates match. I have a fixed point here, and I have a fixed point at 0. At all these 3 points, the rates of these production and degradation terms cancel each other out, right? And so again, the way we want to visualize this is by drawing that kind of a flow diagram. Again, just to make things quicker, I won't be very careful about the magnitude of the arrows, because what I'm interested in showing is their direction. Okay, sorry, maybe I should have aligned these two axes the same way, but this is P1 equals 0, and these 3 points, so maybe I'll make them bigger. These 3 points are exactly these 3 fixed points where the rate of production and degradation match. Okay, so now we have something that's different already. First of all, we have 2 stable fixed points, and I won't go through it, but if you look at these functions carefully, you will figure out that near 0 and near this fixed point, these things will be stable, which means if you move away from them, you return to them. And this middle fixed point is what's called an unstable fixed point. Right? So it's an unstable fixed point. If you move away from it, you're going to flow either to this or to this stable fixed point. So this is all at alpha E equals 0. So in this system where I have auto induction, a gene regulates its own activity, if there's no external signal, the system already has 2 possible fixed points, two types that it can take, if you like, 2 final concentrations of protein where it will reach. One is for 0 concentration of the protein, the other is at some high concentration of the protein. And whether it will go to one fixed point or the other is decided by this unstable fixed point, which divides my total space into 2 regions, right? And again, these ideas are going to become more important when we discuss these things abstractly. I have an unstable fixed point which has divided my whole space into one region where things all flow here, into another region where things all flow here, right? And in general, I have 2 sorts of solutions possible here for what I get as my final cell type. Yeah.
Say a sort of pluripotent state, and it can divide into 2 stable states like the 2 lineages.
Yeah, I mean, so one should be, yes, I mean that's an analogy which will fit. Except of course I'm going to exactly come into how we think about ideas like that, where you could have, as you said, a pluripotent state, and then you have at that particular decision 2 possible differentiated cell types. Here of course I'm doing it for a particular example, but you're right, that's the idea to have. Those are the analogies to have in your mind. Yes. Okay, so now what happens if we increase the value of alpha E? There's only one colored chalk, right? Or as in there's only white chalk. That's okay, but I'm just checking. No, it's okay, it's fine. So let me increase the value of alpha E a little bit. I have to make sure I can draw this. I've not drawn this very well, sorry. Okay, so this first curve was at alpha E equals 0, and now I'm going to take alpha E going up just a little bit. Alpha E is small, right? Again, the actual values don't matter so much. You know that the line has to have the same slope because the slope is just given by gamma. Alpha E is just the intercept, right? At P1 equals 0, positive alpha E will give negative intercept. So you know that the things have to shift downwards. Okay, so as I've increased alpha E, things have gone downwards. I still have 3 fixed points, right? And I can draw my, so I'll call this, so this was 1, this I'll call 2. And I'll again draw the flow diagram. So now I no longer have any fixed point at 0 because things have moved away from 0. I still have my unstable fixed point. Oh, thanks. And I still have this fixed point here. Right, so this is situation 2. This is situation 1. This was at alpha equals 0, no external signal. As I have now increased my external signal slightly, again the idea you should have in your head is as I've increased the concentration of a signaling protein, as I've increased the concentration of some hormone, you know, what is happening to my, what is happening to the possibilities for this protein concentration. And in this case, you see that nothing much has happened, but these fixed points have moved a little bit. I still have one stable fixed point at low concentration of protein and one stable fixed point at high concentration of protein. Now, if I increase the value of alpha E a little bit more, which I'll now draw with a different color. Okay, so alpha E is going up as I keep increasing the value of alpha E. My negative intercept is going to keep increasing. You'll see what has happened now is that I've lost two of these fixed points. Right? There is only one point where minus alpha E plus gamma P intersects with the function f of P, and that's at this high concentration. So what that means in terms of the flow diagram, which maybe I'll draw in the same color. Is that I have no evidence anymore. I mean, technically speaking, there is still some evidence, but we won't talk about that. But in really, I don't have any stable fixed points in low protein concentrations. I just have one stable fixed point at high protein concentration. So if you think about this now in terms of actually doing an experiment, what that would mean is, suppose initially I was measuring low concentrations of the protein and I was increasing my external signal, right? Initially, nothing much would happen. You know, maybe my concentration of the protein would slightly increase. I would be here, but then at a certain point, at a certain value of alpha E, suddenly my protein concentration will jump, and I'll get a very high concentration of the protein. So this thing is called a bifurcation. Okay, and what is meant by a bifurcation is that it is a qualitative change in the dynamics, right? So earlier we were talking about small quantitative changes, which are things like shifting the values of the fixed points, but a qualitative change is something like this, where actually two of the fixed points have disappeared, right? An unstable fixed point and stable fixed point, it's usually said annihilate each other, and you only are left with one stable fixed point. Yeah.
By looking at these qualitative features such as bifurcation,
Yeah.
In this case, like, is it possible to infer if it is by increasing alpha or by varying the effective dilution rate?
You're saying, could I also change this?
Yes.
Could I change the gamma P? So that's an interesting point, and in fact, in some ways, you know, the mathematics is sort of agnostic to which particular parameter you choose to vary. In this case, just changing gamma, if you keep alpha 0, could do something if you just make a low enough slope where, right, you again miss this. But there's a little bit of an issue with that, which the problem is that as long as you have finite gamma, since this function saturates, eventually you're going to hit the function. So gamma is not something you can quite use. But in general, you're very right that you could change these forms in different ways. You could change different parameters here, different things you can control, which can both lead to a certain type of bifurcation. Yeah. Yeah. Yes, yes, yes. So sorry. Yes, yes. Right. So actually there's something you can vary, which is a parameter. So these things are parameters, and then what you observe is changes in behavior. Okay, so time has passed by much more quickly than I thought it would. So let me now at least start taking these ideas and trying to tell you how we want to think about them more abstractly. And then maybe I'll continue this a little bit more tomorrow. Let me see. Okay, so essentially what the mathematics does, and again I'm not going to give any formal mathematical discussion here, but at least I want to give you an overview of how a mathematician would deal with a situation like this. So I've already shown you a sort of you can have certain types of cells, certain which we correspond here to these stable fixed points. We've not actually discussed how these would give cell types, but I'm at least making an analogy for you between stable fixed points and cell types, and cell state and sort of the concentrations of proteins in the cell itself. The issue, of course, is that there are many, many proteins in the cell. The more we seem to discover things about how cells take decisions, the more complex it seems to get. So if you take a typical signaling pathway, which is the way that often cells make decisions, these signaling pathways now have tens of components, many, many components. And as I said to you before, you don't quite know how to write down the equations for these components. So you want somehow a more abstract formalism where you don't want to write down the precise equations of motion, but still you want to be able to understand experimental results, like the kind that if I increase a certain signal, if I increase the concentration of a signaling protein, or if I put in a hormone at higher concentrations, I start getting different phenotypes. My cells, I start getting one type of cell or the other type of cell. If I have two types of differentiated cells, as I increase something that I can experimentally control, it changes how many cells end up becoming one type versus the other type. But you want to do all of that without necessarily knowing what is at the basis of the mathematical equations that are at the basis of the change. And that's really the point of view that mathematicians take. And again, I won't give you the formal point of view, but at least I want to give you the ideas behind it. The first important idea is the idea of what's called topological equivalence. So again, now I'm going into sort of a more formal part, but hopefully I'll get all of you to appreciate the ideas here. So the idea of topological equivalence is that really, I know certain qualitative things about my equation, but maybe I don't really know exactly what what the equation is. So how do I try and formalize this idea that we've been discussing for some time, this idea of qualitative behavior, right? And so the things that are qualitative, as you saw, were the number of fixed points, the type of fixed point, whether it's stable or unstable. These are all qualitative parts of the flow. What is quantitative is these equations, right? And so you want to go away from this kind of quantitative description to a more qualitative description. So to give you an example of how two things will be qualitatively similar, right? Consider, and now I'm going to write in indices. Suppose you had a certain whatever circuit where many, many proteins were involved, and you wrote down a certain equation for it, pi dot equals fi of p. Fi are certain functions. We're not going to write again, we're not going to specify what those functions are. Okay, now suppose I tell you I have another equation, which I'm going to call a different variable. And I have a map. So whenever I don't write the indices, it means that it can depend on all of the i's. When I write the indices, it is to show that I'm talking about a particular I, right? I can derive from this the conditions under which these two equations are right. So if I'm given a map like this, then I can tell you how my equation for y can be written in the equation for p. So sorry, this is not x, this is p. Okay, so let me be a bit clearer. Okay, so I have an equation for p, I'm giving you another equation for y, and I'm telling you that y actually depends on P with some arbitrary function H, right? Then I can write down an equation for Y. Okay, and this thing I can call, oh sorry, I used P there and X in my notes, that's why I keep messing this up. And this thing I can call okay, so again, to tell you the broadly what I'm doing, what I'm saying is that I choose to write down equations in certain coordinates, protein concentrations. In principle, you want to think abstractly. You want to think about the possibility of changing the coordinates in which you're understanding the system, right? And one way to do a general coordinate change is to write down new coordinates yi, which are some functions of your original coordinates pi. And if you do that, right, you can then derive the equations in those new coordinates yi. And again, this is sort of abstract. You can ask why you'd want to do this, which hopefully I'll convince you this is a useful thing to do. Let me again see how much time I have. Yeah.
Will that have a dou G over dou H term as well?
Where?
In the equation where you have changed coordinates.
So maybe I skipped a step. So yi is equal to hi of p. That means yi dot, right, d by dt of yi is d by dt of hi, which is pi of pi dot. Right, so this is just a chain rule.
Thanks.
But you're right, there is a G there. And so exactly what I want to say is that if summation k del hi by del pk, pk dot is equal to G of h, so this is Gi of h of p, then these two equations are essentially equivalent. Then really this first equation that I wrote down, given this coordinate transformation, is exactly the same as this equation is exactly the same as this equation. The only thing is I'm looking at it in different coordinates. And this abstraction of really thinking about the space of all coordinates that I could choose, all ways I could choose to describe my system, is very, very useful. It takes you away from a specific description and allows you to think about qualitative ideas. And hopefully next, tomorrow I'll show you how this kind of a description is more useful when you don't know actually the equations at the molecular level. Okay. So I won't show you for want of time, but you can calculate it. It's not a very hard exercise if you've done these kinds of calculations in the past, to see that yi will have exactly the same fixed points as pi, that gi of, for every fixed point here, there will be a fixed point here. And in fact, not just the fixed point, but also the linearization near the fixed point will be the same, right? And so linearization near the fixed point, meaning that in general these are nonlinear equations. Yeah. Yeah. Between y and pi, no, it doesn't. No, it doesn't. So there are equations that will be different. If this condition is satisfied, then these equations are equivalent. That's what I'm saying. And in fact, you need to have assumptions on the map. So here I'm actually assuming that's a diffeomorphism, but I'm not. You need certain assumptions to satisfy certain other properties. But in general, if you have a map which satisfies equations like these, it means that the two equations, this one and this one, are qualitatively the same. And this is this idea of topological equivalence. And what this means is that the fixed points have to be the same, right? So if I have two stable fixed points here and one unstable fixed point, my new equations also need to have two stable fixed point and one unstable fixed point. They don't need to be at the same position. In my yi, they could be in some other values of yi. But I still need to have the same, same three fixed points. It turns out for certain maps, which again we won't go into mathematical assumptions, but technically speaking for diffeomorphisms, the eigenvalues are also the same, which means the linearization near these fixed points, the flows are in general non-linear, but you can linearize them near a fixed point. These linearizations are different, but the eigenvalues of the matrix, the eigenvalues of the linearization are also the same. Which of course means if I have a stable fixed point in one set of equations, I'll have a stable fixed point in the other set of equations. Yeah.
So just a naive question. So this is it something similar to what we know as topological conjugacy in dynamical systems? For example, the logistic map and the tent map, they
Right.
are topologically conjugate.
So it's a similar idea. I mean, so this is for a continuous dynamical
for a
system. You can
continuous
build up exactly the same idea for maps. Okay, so essentially, so let me, I mean, instead of going too much into the mathematics, let me just tell you why I want to think about this idea of topological equivalence. And essentially, I want to think about it because in a biological system, which is like the one somebody described, we have a pluripotent cell that's differentiating. What I do know about the system is that, just observing it phenotypically, I know that there are two types of cells, that it can differentiate into in that time period. Which means that I know whatever the intrinsic dynamical system is, it will have two sorts of, it will have two stable fixed points. The approach that dynamical systems theory takes is that instead of trying to write down the equations which produce those fixed points in terms of specific proteins, let me, look at the class of all systems which will have two types of, for example, stable fixed points. And then let me write down equations which are as simple as possible, which will have two fixed points, and then try and model my experimental system using those equations. So again, it's a different kind of approach. Instead of now writing down equations for quantities that you know are there, instead you look for what is the qualitative behavior. Once you know the qualitative behavior, you say, let me write down the simplest equations which have this qualitative behavior, and then let me try and fit it to experimental data. So there's one final thing that I want to tell you about. So it's 1 o'clock, but maybe I'll just take a few minutes. And that is this idea of bifurcations, which I've now erased. So if I have, so first I've now set up the idea of qualitatively equivalent systems using this idea of topological equivalence. What a bifurcation does is precisely produce a topologically non-equivalent system, And what I can do biologically, as I'm varying my signals, is that I can produce systems that are topologically non-equivalent. And essentially the big, sort of the task of dynamical systems theory is to draw out bifurcation diagrams, where as you vary a certain parameter, you just like we did in this example, as I vary a certain example, this is a certain specific system. As I vary a certain parameter, I went from a system which had 3 fixed points to a system that had just 1 stable fixed point. The mathematical approach is essentially that I want to tell you in your system, if you tell me a system and tell me that it has, say, 3 stable fixed points, I want to tell you that if I take any parameter in the system, as long as you're just varying one parameter, what are the ways in which that system can change topologically? So again, it's an abstraction. Instead of writing down specific equations and saying how, as I vary a parameter, the system changes, you think about the space of all systems which have behavior like this, the space of all systems which have behavior like this, and you try and write down the simplest system which shows the behavior transitioning from a system like this to a system like this. And tomorrow what I'm going to at least talk about in the first half, the second half maybe I'll talk about something slightly different, is to show you how these abstract ideas can be used as a tool to make sort of concrete quantitative models of how cells take particular decisions in early mouse development. So maybe I'll leave it at that because we're out of time. Yeah.
Two topologically equivalent systems
Yeah.
will exhibit the same bifurcation diagram.
So topological equivalence, bifurcation by itself, so bifurcation diagrams distinguish topologically inequivalent systems. So really what you want to do in a bifurcation diagram is, write down a parameter and say, as I vary this parameter, P, all of these systems, for example, will have a flow diagram like this. All of these systems will have a flow diagram just like this. Okay, now where these fixed points are and so on, all of that will change in this region. But the point of the bifurcation diagram is to tell you that this region and this region are fundamentally dissimilar. And all of these are topologically equivalent systems, all of these are topologically equivalent systems. And a bifurcation diagram partitions your space into different systems that are topologically equivalent. And of course, this is an example where you can just change one parameter, right? So the other important thing that happens is you don't care what the parameter is mathematically. It could be decay rate, it could be external signal. The important thing is how does it change the qualitative nature of the flow.
So you can talk about topological equivalence only within a particular regime such that it does not cross bifurcation point.
That's what the bifurcation diagram precisely does. Yeah, it partitions spaces which are topologically equivalent. Yeah, that's precisely this function. Yeah.
I think that point connects with what Amitabh was talking about topological conjugacy, what you are referring to. So Amitabh gave this example that any smooth unimodal map will have this period doubling bifurcation diagram, characteristic of logistic map. You take any smooth unimodal map, they will show exactly the same bifurcation diagram. Okay. So what you're referring to is actually this topological conjugacy of systems. I think the question was like, would two systems exhibit the same bifurcation diagram, as in like period doubling bifurcation? The universality.
Oh, right. Okay, I mean, yeah, I mean, okay. So then you're really talking about the space of all bifurcation diagrams. So that's okay. So that's interesting. I mean, so you're right. So in this case of period doubling, yeah, there's something where you can get a universality through these unimodal maps. It's a slightly more complex question what you're saying then. I didn't realize that. But what you're saying then is a slightly more complex question, which in general there are no general results for this. In specific cases you can get results for, are there bifurcation diagrams? You can start thinking about the space of bifurcation, space of systems which show the same bifurcation diagram. Yeah, these general results are harder to get, but I'll show you actually tomorrow also. With certain assumptions, you can get spaces which also have the same bifurcation diagram, not just, I didn't realize that was a question. Yeah, so that's a good question, and again, there are no general results. There are certain cases where you can make some claims about this, yeah. Yeah, there are two. I think there was a question at the back also.
So about this map that takes Pi to Yi, right?
Yes.
So does it have any properties, like does it have to always be invertible and?
Yes, exactly, yeah. So in fact, for topological equivalence, you just need a homomorphism. It needs to be invertible and one-to-one. In this particular case, I've actually, to do all of this, I was thinking about diffeomorphism. You also need the derivatives to exist. and also you need the inverse of the, so you need certain more assumptions. But yes, for topological equivalence, strictly speaking, you need just invertible and one-to-one. Yeah. Sandeep, yeah. 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 So it's, I mean, I would say that's a research question. I don't know if there's any answer I can give, but I mean, they seem to have similar flavors, I would say. I mean, there has been, in a different context, I mean, what I'm doing here would roughly come under what's called normal form theory in dynamical systems. There have been equivalences between the renormalization group and normal form theory that people have tried to make. But in this case, I mean, this I know there are no real results. I mean, the sloppy models approach, so for people who don't know, the sloppy models idea was that if I now actually write down, take a complex system, I write down all the equations, and then I study the behavior, if I study some output behavior, and I look at its dependence on parameters of these equations, turns out that it doesn't really depend on all of the parameters of the equations very sensitively. I think that's something you would expect also from this kind of an approach. But the harder thing, the thing that's ignored here is how do you do the dimensional reduction, right? So that's the harder question. How do you do the dimensional reduction from a very large space to a small space which has the same behavior? The approach I'm taking is that you know the dimensional reduction exists, so just model the behavior in the small space. The Setna thing was trying to say that, again, the dimensional reduction should exist. How exactly to do the dimensional reduction is a harder question, and some people have tried, but it's typically, nothing very, very satisfactory, I would say, has come out yet.
Yeah, something like related to this. So one, you said that doing some kind of a renormalization where you get rid of the irrelevant parameters.
Yes.
But what about something like taking some kind of a Poincare section or trying to, from this many dimensional system, find a map which actually captures the essential dynamics in a much reduced dimension? Is it possible?
Well, so Poincare section you typically do when there are oscillations, right? Then you would take, I mean, in a sense, this was, if I should say, in a sense, this was Poincare's program. This was really, this idea, something that Poincare was really, I would say, started. Poincare wanted this idea that there should be normal forms of dynamical systems. Yeah, I mean, there is, so I don't know, maybe I didn't answer the question, but what was the?
No, I mean, I was thinking of like the conventional Poincare section that like
you take the dynamics and you take a section and then you see a map on the section is effectively giving you the dynamics.
Yes. So.
Yeah, I mean, so yeah, again, this is, I think, for specific systems, this can be very useful to do. Yeah, again, these are, so the idea, I mean, here the thing is, these are all very general results, right? They're all, yeah, but yeah, I agree, for specific systems, there may be things that are very useful to do, yes.
Okay, so with that, let's thank Chishpan. Okay, we now break for lunch, and lunch, as always, is across the road on the upper floor of the campus.